Thank you, Oscar, for that very nice introduction. Without further ado, I would like to start presenting the problem on which our company is working on, which is the following. The problem here is that most modern drugs work on the principle of inhibition, where you put a small molecule inside what we call an active site cavity. The active site cavity is where a particular protein that we are interested in inhibiting expresses its activity. And we put a small molecule that goes and settles into this site, which results in an inhibition of that protein's activity. And this is the principle behind a majority of modern day drugs. Now, the unfortunate case is that there's only 10 to 20% of all pathogenic proteins that can be inhibited in such a manner. And so what do we do to go beyond this set of proteins? And the solution that our company explores is that of degradation. We make use of existing mechanisms within the cell. So it's amazing. The cell has mechanisms for degrading proteins which have come through evolution to dispose of proteins that are either malformed or have outlived their usefulness. And our strategy here is to make use of this machinery to dispose of proteins that cannot be targeted by drugs. This approach is known as targeted protein degradation and is demonstrated on the next slide. So we can see here that the E3 ligase complex, which is responsible for the degradation of enzymes within the cell, degradation of proteins within the cell, gets attached to the protein of interest here and tags it with a molecule known as ubiquitin. The ubiquitin tagged protein of interest is then recruited by what we call the proteasomal degradation enzyme, which proceeds to dismantle the protein into harmless peptides. This entire system is called the UPS, or the ubiquitin-proteasome system. So where is our innovation in this? The method that we are trying to employ to cure diseases is design molecules known as degraders. Degraders are molecules that perform a very important function, namely they attach the E3 ligase, which does the degrading, to the protein of interest, which we want degraded. And this attachment is done by means of interacting ligands. So you have a ligand that interacts with the E3 ligase, and you have a ligand interacting with the protein of interest. And you have a linker that binds these together and causes the protein of interest to interact with the E3 ligase. And then proceeding, once we have a successful interaction, then the E3 ligase can proceed to degrade the protein of interest. The advantage of this approach of designing degrader molecules is that it offers stability. The molecules themselves are stable, and it allows us to target a large class of proteins that are inaccessible by means of inhibition. We also work directly on proteins, so we do not need to modify the DNA. We can cure diseases, so to speak, rather than prevent them. They're much more selective in their approach, meaning that they have potentially lower toxicity, and also they require lower dosage. So the mechanism of degradation and the action of the degrader molecules is showcased in the next slide, where we have a video. I would request each person to click on the video, as this is not something that starts when I click on it.
Arjun? Sorry, Arjun? You're mute, we can't hear you.
Hello. Sorry. I hope everyone has had an opportunity to see the video. And as the video shows, our company's thing is to design these degrader molecules. I will continue to the next slide where we ask the most important question here, as in, if this is so awesome, then why isn't everybody doing this? Why aren't degraders the thing on the market at the moment? And the reason is it's difficult, just very simple reason here. And what is difficult about this thing is what I would like to mention here. So the difficulty is that the molecule in question, namely the degrader, and specifically the heterobifunctional degrader, is a molecule that consists of two parts, the ligands here that interact with the proteins, as well as a linker. And the issue with a molecule of this complexity is that the interaction of this molecule with the respective proteins and the eventual degradation of the protein depends on several biomolecular interactions. So firstly, we have the interactions of the fragments with the respective proteins. And then equally important, I would say, if not more, is the interaction between the proteins themselves in the presence of this degrader, as well as the constraints enforced by the presence of this linker. So this linker severely constrains the possible interactions that can happen between both the proteins, as well as the interactions that can happen between the ligands and the proteins. And in this large space of interactions, typical methods of simulating these drugs or doing an in silico validation of these drugs using either molecular dynamic simulations or docking-based Monte Carlo simulations are very inefficient. And this leads us to take the following approach to solving this problem. So enter deep learning. It's the magic word that everybody seems to be talking about these days. For those unfamiliar, deep learning can be thought of as a means to map a certain set of inputs to a certain set of outputs. At its fundamental, that is it. And the versatility of deep learning lies in the fact that the inputs and outputs can take any range of forms. And this intermediate set of neurons and connections that you see are also as varied as they come. So the entire field of deep learning thrives on the possibilities that this very fundamental scheme allows us. And what do we use this for? So we use deep learning to predict the biomolecular interactions within the degrader complex. As mentioned a couple of slides ago, the difficulty in designing degraders is primarily to predict the interactions that are involved in that. And instead of using the traditional cheminformatics-based methods, which are very, very computationally intensive, we find that the use of deep learning architectures can leverage the availability of data to have algorithms that are incomparably faster to the traditional approaches, which makes this a significant advantage or a significantly worthwhile investment in terms of solving this problem. Additionally, we have developed a novel protocol that makes use of a machine learning technique known as Bayesian optimization. And this Bayesian optimization enforces the linker constraints that we have talked about earlier. And I will proceed to give you an introduction to our pipeline, the pipeline that we are working on to solve design degraders. The first step in our pipeline is a protein structure determination, where we determine the structures of the proteins that we care about. In this case, these proteins would be the E3 ligase, as well as the pathogenic protein for which we would like to degrade. This is not necessarily something we need to do in-house, as for the diseases that we are interested in, the responsible proteins have already been studied, and we have like existing X-ray crystallography structures of these, which we can make use of. The second part of this pipeline is where we begin to leverage our in-house models. And this part involves the protein ligand interaction, as in where we choose the ligands that we would like to be that we would like the degrader to be composed of. And how do we do this? By moving through a database and selecting the ligands that have a high prediction score corresponding to the protein that we're interested in. And this becomes extremely feasible because of our extremely fast models that we have which use deep learning. Once we decide the binding fragments or the binding ligands for both the proteins and we calculate the combined structure, we use this combined structures for the next step, which involves us filtering through the vast space of protein-protein interactions. So there's a large possibility in the space of protein-protein interactions that we filter through by enforcing not just the constraint of the fact that the proteins need to interact energetically favorably, but also the constraint that they need to interact in a manner that makes it feasible to attach a linker to the ligands, right? So we would not be interested in considering interactions if the two fragments are extremely far apart in whatever orientation the proteins choose to interact. So these would become infeasible in the presence of the linker. So we take into account these linker constraints in this loop. And the Bayesian optimization loop allows for a systematic means to do that. Once we have refined this large space of protein-protein interactions to a small candidate set of possible interactions, we then conclude this process through the final refinement step, which takes the small number of possible protein-protein interactions that we have filtered out and runs it through traditional cheminformatics-based methods such as Monte Carlo simulations and or molecular dynamics simulations to finally arrive at the final structure of the ternary complex. And this structure of the ternary complex is one of the, I would say, one of the objectives of our entire pipeline, because this is what will enable us to predict whether or not a given degrader is effective, right? So additionally, I would like to mention that in step 3, we have a deep linker generation where we generate a linker given the context of the particular protein-protein interaction that we are seeking to optimize. So ultimately, from start to end, we have the protein to be degraded. We choose the ligands, we choose the linker, and then we are able to estimate or determine the ternary complex that is formed by the generated linker. So this is the set of works that form the state of the art, as in we compare our works to these works. And I will pause here for a moment for anybody interested to have a look. So my next set of slides will discuss the modules that we have used. And I would just like to recapitulate what modules I've introduced. We have a deep interaction prediction module, which enables us to predict the interactions of the ligands with the proteins, as well as the interactions of the proteins with the proteins. We have the deep linker generation module, which is able to design the linker for any particular geometry of ligands or any particular set of constraints that we may impose on it. And we have a deep conformation generation. We're working on a deep conformation generation module, which will enable us to study the structure of the generated degrader. So the first module that I would like to introduce is the deep interaction prediction module. And as the name suggests, its job is to predict the interactions, either between the protein and the fragment, which is shown in the figure here, or between a protein and a protein. This model acts on what are known as graphs, which are certain mathematical constructs. So a graph is a mathematical construct that consists of nodes, which are connected together by connections we call edges. And this sort of a mathematical object is very conducive to representing the properties of molecules, because as you can imagine, simple molecules, for instance, can be represented by graphs where the atoms are nodes and the bonds between these atoms are the connections. And for more complex molecules and larger molecules, such as proteins, we understand that the geometry of the proteins plays an important role, which allows us to use methods that represent the protein in terms of what we call as surface graphs or geometry-based graphs, in a sense that these graphs represent the geometric properties of the protein. And we find that these methods are very successful in predicting the interactions that we're interested in. We use deep networks that operate on graphs. There's a large literature of deep networks that operate on graphs, which we make use of in order to predict the interactions from the graph data that we have. This deep interaction prediction module is something that plays a role in the steps 2 and 3 of our pipeline, which involve the protein ligand and protein-protein interactions, respectively. Our current results on this are very encouraging. We match the performance of an existing state-of-the-art method termed as massive. And we have a significantly improved time complexity performance. As you can see, nearly a 50% time we require in terms of things. And I think our most important advantage here is generalizability that owes to the efforts of our teammates to develop a very varied database of proteins to train the deep network. As we know, deep learning is all about the data used to train it. And we train our models on a very diverse data set of proteins as compared to the data set used to train the state-of-the-art massive model, which allows for generalizability to a large range of proteins. The drug-target interaction, unfortunately, is not yet publicly disclosable, and we will be able to show our results by November. We proceed next to the Bayesian optimization loop, which forms the backbone of predicting the protein-protein interactions, as well as the protein complex. Sorry, not the protein interactions, but in order to be able to decide which protein complexes are feasible given the degrader, we use the Bayesian optimization loop. So the basic need for this comes from the fact that the space of protein-protein interactions is very large. And how do we narrow this down? Because we know that proteins are large, flexible molecules. So how do we know what interactions are feasible in the context of the degrader molecule? This problem is something that is made, it's almost like a perfect problem for an approach known as Bayesian optimization. And the logic of Bayesian optimization is rather simple. We have something called as a surrogate model. And the surrogate model basically is trained to estimate a computationally expensive fitness score of sorts. So in this case, we're talking about a fitness score that takes into account not just the protein-protein interaction, but also the constraints imposed by the linker. So this is computationally expensive. So we do not want to be evaluating this term for every possible configuration and then filtering out. So we use a surrogate model to estimate this sort of an expensive fitness score. Additionally, we use an acquisition strategy, which takes the fitness values that are given by the surrogate model and intelligently chooses the next samples for which we calculate the actual fitness. And then we update the surrogate model, and this continues as a loop. So this is an iteratively, this improves the quality of the samples that we have in an iterative manner, while requiring us to calculate the fitness values for very few points. So this is the basic principle of Bayesian optimization. Before I go into detail regarding the fitness, I would like to just show people the effectiveness of Bayesian optimization in modeling a fitness score, which you can see here. So the map on the top is the fitness score that is a true fitness score for a certain subset of the space, whereas the map at the bottom is that predicted by the surrogate model. And the fitness score over here is the fitness of a docking function. And we can see that we have a function that can predict this docking fitness to very high accuracy. And this function has been trained using very, very few samples, a few hundred samples, using which it allows it gains this level of accuracy. So making Bayesian optimization a very useful technique. But now the question is that this is all well and good. What are we trying to optimize, right? And that is something that I'm coming to at the moment. The first constraint that we care about when two proteins interact is the PPI fitness, standing for the protein-protein interaction fitness. And as mentioned previously, this is calculated using the deep interaction prediction module that I have just talked about. So this gives a measure of how strongly two proteins can interact given a particular orientation. As you can see, each configuration is represented by some kind of orientation of the two proteins. So given a particular orientation, how strongly do the proteins interact in this orientation is something that is given to us by the PPI fitness. The additional fitness, and this is the most important fitness, given that we're talking about degraders, is what we call the constraint fitness. And in simple terms, this evaluates the feasibility of linking the ligands with a linker. So for any particular orientation of these proteins, we also know that the ligands will have a certain relative orientation because they're bound to their respective binding pockets. And given this orientation, is it possible or is it not possible to connect these ligands together with a valid linker? This is the constraint that we seek to model. So for instance, like I would say that something like this would be infeasible, right? If you can see the pointer, it's a constraint, a protein-protein configuration like this one would be infeasible because of the fact that the binding pockets are extremely far apart. So you wouldn't, this is not a configuration that would be feasible in the context of a degrader. So in order to do this, we make use of the following modules, namely the deep linker generation, which generates a linker for the given configuration, as well as the deep molecular conformation generation, which then analyzes the geometry of the generated degrader to see if it is consistent with the configuration. And based on this, we have a score that tells us whether any particular configuration is feasible. I will proceed to describe the deep linker generation and the deep molecular conformation generation. We see here that the deep linker generation, in terms of its name, its job is to generate a linker to join the two fragments that bind to the proteins. And we do this by means of a variational autoencoder, which is a standard generative model in machine learning, in deep learning. And the input to this model is the two fragments in their respective positions given a particular protein-protein configuration. Right. So given the orientation of the two proteins, we also know the orientation of the fragments as they are bound to these proteins, right? And these orientations are fed as input to the algorithm. And by means of a variational autoencoder, the algorithm generates the graph of a linker that you can see over here. So on this side, we can see that taking into account this geometry, the deep linker generation has generated this graph. And this graph is generated taking into account this geometry and potentially taking into account pharmaceutical properties. And this is only limited by the availability of data. The model in itself allows for us to make use of any available data regarding preferable molecular properties, namely solubility, toxicity, drug likeness. So any property for which we have data, we can include into the model and generate linkers that optimize for that. So we take into account the geometric constraints here, as well as any pharmaceutical biases that we have introduced into the model, and generates a graph. Then in order to validate this graph, we find the conformation, we find the 3D structure of this generated graph that we find over here, and see if it matches up to the original input orientations. If it does, then it means that we now have a valid linker that can join the two fragments as provided here, right? Which will give a high constraint fitness to this particular protein configuration. So the fitness of the generated molecule feeds into the Bayesian optimization loop as the constraint fitness. The deep linker generation, we have very encouraging results for the deep linker generation. And the reason is that compared to a state-of-the-art method, namely D-linker, we use an advanced coordinate system to incorporate more of the geometrical information of the fragments. And this enables us to recover valid, both pharmaceutically valid as well as just valid in terms of the data linkers compared to D-linker. So we see that we don't lose anything in terms of novelty. We have a much higher recovered percentage, and we are very comparable when it comes to the pharmaceutical filters, including, like, for instance, the synthetic accessibility, the ring filter, as well as the Payne's filter. And this means that the additional inclusion of geometric information that comes from the protein configuration that we're analyzing at the moment is something that significantly improves the linker generation, which has made our approach particularly effective. Our final model here is the deep molecular conformation generation. So in terms of the deep linker generation, we know that there is a final procedure to that where we calculate the 3D structure of the finally generated degrader. And we choose to do this again through the means of deep learning, primarily because degraders are very large molecules, and generating accurate conformers by means of cheminformatics methods is something that is computationally expensive. And the deep learning method that we choose generates a conformer of a degrader given a graph, and it does this through the means of a force function that deforms a random initialization. So you have a molecular graph, you have a random initialization of the coordinates of the atoms, and then the algorithm itself has learned an internal force field that can deform this molecule to become a valid conformer. And how does it know what a valid conformer is and what this force field is simply that it learns the distribution of data. So we have a large amount of data in terms of what various molecules are and what their 3D structure, what the potential 3D structures are for these molecules. And this model can learn a statistical model of what a good molecule is. And it can deform any initial coordinate to be that. And the data for this model, for instance, can be generated through more computationally expensive means to get more accurate conformers. But once we train the network, then inference for a new molecule will be absolutely rapid. Now, to summarize our approach and put it together so that we know how each of these modules come together, to recapitulate the pipeline. The input to our model is the structure and sequence of the E3 ligase or the protein of interest. And optionally, if we would like to test specific ligands, then the ligands can form an input to the model as well. The process of our pipeline is as follows, where at first we determine the ligands if not provided, and we determine the protein ligand interactions and the complexes. We then proceed to determine all possible protein-protein configurations or possible protein-protein complexes that are valid in the context of the degrader, in the sense that are valid and can be linked by a valid linker and have a strong interaction. And this makes use of all of our pipelines: Bayesian optimization for the backbone, deep interaction prediction for the protein-protein interaction, as well as deep linker generation and molecular conformation that form the basis of the constraint score. The final step in this is that once we have filtered the space of possible protein-protein complexes to very few candidates that are now valid in the context of both their interactions as well as the constraints, we can now refine these configurations using either molecular dynamic simulations or something that is like cheminformatics based, using a traditional force field or more biologically oriented approaches. So at the end of this, we expect to have the graph of a generated heterobifunctional degrader. Namely, we have two ligands which interact with the respective proteins, and we have the linker. Along with this, we of course expect to have the structure of the ternary complex that is formed by the E3 ligase, because this is the complex that is going to enable the E3 ligase to tag the protein with the ubiquitin. And only if we can understand the interaction between the E3 ligase and the protein can we begin to predict the activity of the generated degrader. So these are the outputs of our model. And I think this is something which must be on all of your minds right now, as in I have mentioned 5 deep learning modules, thrown a bunch of jargon, and why do any of this, right? Well, in terms of advantages, the easiest advantage to sell here is a tremendous speed boost. So as a simple example, the deep interaction prediction is around 100,000 times faster than a traditional docking-based approach if you would like to calculate the PPI fitness, for instance, right? So calculating the PPI fitness is something that takes in the order of several seconds, and like it's something that and maybe even minutes if you have a more complex docking scheme, right? And also for protein ligand fitnesses, and this allows us to incorporate very novel workflows. Like for instance, we can perform a very rapid search for searching each ligand. We can perform a Bayesian optimization loop based on the PPI fitness, and we can like basically test out a lot more possibilities, just to put things simply. Right. The deep linker generation, in addition, allows us to encode several constraints, including the geometric and pharmaceutical constraints in the generated linkers. Usually this is something that we would relegate to the human in the loop with their enormous knowledge of just what functional groups are acceptable, what functional groups are not, what kind of compounds are toxic, and things like that. Or even like what kinds of compounds can join two molecules at a specific position. All of these things are possible. It's possible to encode this information within the deep linker generation, and allows us to give a much more informative constraint score for our Bayesian optimization. And the Bayesian optimization itself allows us a means to perform a very efficient filtering of possibilities, and it is something that can make use of a variety of fitness scores, a variety of neural network architectures, as long as it has a fitness function. So this is a very principled means of going about the filtering in a very computationally efficient manner. And to answer the question of why do all of this, I think it's important to say that we as a company do all of this, at least with the intent of hopefully ending many of the diseases that plague us today. And we hope to be able to serve humanity in this way. In that respect, I would like to introduce Celeris's business workflow, where we are building the following 3 modules, and they have the names of Xanthos, Hephaestus, and Hades. The current model that I have presented is Xanthos, which is the model that does in silico degrader generation and ternary complex prediction, and all in silico validation methods come under Xanthos. We are currently actively involved in the Hephaestus and Hades part of the plan, and we are constructing a lab that has in St. Peter, Graz, at the moment, with a very large area and with an automated lab that will enable us to perform tests on the compounds that we generate using Xanthos. Hades is where we actually conduct the tests inside cells, and we are able to feed the results of those tests back into Xanthos to be able to improve our algorithms and generate better molecules, potentially using an active learning pipeline or a Bayesian optimization pipeline. And to talk about results, we have We have a series of compounds that we have already submitted for patenting. We have a patent file for these compounds, out of which we have chosen 3. Of course, we have blocked out parts which we cannot show yet. And we are already in the process of synthesizing these compounds and hopefully being able to take that insight into our next set of algorithmic improvements. I would like to thank all of my team members for making this work possible, and thank all of the audience members for taking their time to be here with us today. Thank you.